I'd like to begin just by introducing our speakers before I turn it over to the team for their presentation. The speakers here are all work in this student collaborative organic plant breeding project, and they include Will Hazzard, who is a second-year master's student with Tom Gradziel in the peach and almond breeding program and was previously a research assistant in the breeding program. His research focuses on epigenetic inheritance of genetic diseases in almonds and variation in DNA methylation in different tissues in almond. Before coming to Davis, Will was a legislative aide to Senator Maria Cantwell of Washington State with an emphasis on agricultural policy. We also have Antonia Palkovic, who's worked on several farms in upstate New York before coming to California to study agriculture. She also farmed vegetables organically in Washington State and Minnesota before returning to California, where she currently works as a field-based plant breeder at UC Davis. Her current breeding projects involve garbanzo, lima, and common dry beans, as well as mentoring graduate and undergraduate students in fresh market, vegetable, and flower breeding for the organic sector. We also have Travis Parker, who is a postdoctoral researcher at UC Davis and has been the lead of the Scope Common Bean breeding team since 2015. His research projects focus on the use of biodiversity and technology to promote sustainable agriculture. We have Laura Rosser, who is a co-coordinator for the Student Collaborative Organic Plant Breeding Education Program at University of California, Davis. She manages the internship program and the Zinnia, tomato, pepper, and wheat breeding programs in Scope. She graduated from UC Davis in 2019 with a BS in Sustainable Agriculture and Food Systems. We have Luis Salazar, who is a PhD candidate at the UC Davis investigating the genetics of salinity tolerance in lettuce. Luis is also the project lead for the tomato breeding program with the Scope project. And finally, we have James Weeks, who is a first-year PhD student at UC Davis and a technician for the Cal Poly Pomona organic tomato breeding project. His past and current projects have focused on improving fruit quality in organically grown tomatoes. The way that this presentation will work is the presenters are going to start with their presentation, and we will have time for questions after their presentation. When it's time for the question and answer period, you can feel free to type your questions in chat, or you can raise your hand using the raise hand emoji, and I will call on you as best I can in order. If you have questions or thoughts that arise during the presentation, feel free to type them in chat, just in case you don't feel like you're going to forget them, and then we'll get to them after the presentations. With that, I would like to welcome Antonia she's going to be starting off this presentation.
All right. Hi, everyone. As Jared said, my name is Antonia Palkovic, and I'm one of the staff coordinators for the Scope project at UC Davis. So I'm just going to give you a brief overview of what the Scope project is. So this was a grant that we got initially in 2016 and then was renewed last year through the USDA OREI grant funding system. And it was an idea that Charlie Brummer, who's a professor here at the UC Davis Plant Breeding Center, and Jared with OSA really came up with together, and we host it mostly at the student farm on the UC Davis campus. And since the renewal of the grant funding, we're also doing some research with Cal Poly Pomona. Next slide, please. Right, so Scopus started with two main goals. The first was to give students more opportunity for hands-on plant breeding education. So at UC Davis, there's a lot of opportunities for classroom learning and to a certain degree, lab education, especially for graduate students. But one thing that we were hearing in feedback from industry is that students really needed to have more hands-on experience actually working with plants. So that's how the education piece came in. And then the second goal that we had was to release varieties that were practically useful for organic farmers, particularly in California. There hasn't really been a lot of industry breeding targeted at organic growers or organic farming systems. And there's been clear research that these farming systems really are unique and have particular problems, such as like different soil fertility cycling than what you might have in a conventional field. Growers are more limited in their abilities sometimes to deal with pests. They often have different markets than conventional growers target. So it makes sense to actually breed for their farm systems in particular. And then finally, we wanted to make these varieties that we come up with publicly available so that anyone can save the seed and sell it or use it to breed with if they have their own breeding interests. So since 2016, we've had over 40 graduates participate in the SCOPE project, including 13 of them acting as team leads, which means they basically got to manage their own breeding project as they might when they become breeders after graduation. So that's been a great hands-on experience for those students. It's given them the opportunity to work with farmers to figure out how to manage field experiments, both in terms of the planning for statistical design and then also, the day-to-day management issues of how to get the crops cultivated and fix irrigation lines. And they've also had the opportunity to go with us on field trips and to present to our undergraduate students and their peers in seminars. And the field aspect of this project, I think, has been really important, especially in the pandemic. It's been a way for graduate students who started in the middle of the pandemic to still be able to connect with their peers in person, which has been fairly difficult over the last year and a half or so. We've also hosted over 80 undergraduate students in the SCOPE project since 2016. And so that gives them the opportunity to learn basic plant breeding principles and to just really develop their research and communication skills, things that they will probably use in their future careers, whether or not they go into plant breeding. And they've also had the opportunity to learn about organic agriculture. And our interns have participated in a regular seminar that we've hosted weekly for pretty much every quarter since the project started. So that's been a lot of presentations. So the crops that we have been working on, we'll go into detail on most of these, but currently we're working on wheat, which we added to the project in 2018. So that's our main winter crop. Tomatoes, which started as like an inbred breeding or OP breeding program in 2016, and now we're kind of shifting into more of a hybrid breeding program. Zinnias we added in 2020. There was grower interest in having improved zinnia varieties, but also it's a nice teaching tool to have something that's really immediately visually interesting for students to look at. Things that we have in the pipeline for release. Travis is going to tell you about his 5 common bean varieties that we released last year. We're working on jalapeno and bell pepper releases, possibly later this year or in 2022, and also a lima bean project that was started early on with SCOPE and kind of neglected for a while, but maybe we'll have some releases soon. And we're always looking for new projects for the future that meet growers' needs and also kind of broaden our educational capacity. So we're thinking about sweet potatoes and possibly Asian specialty vegetables for the future, pending funding. Right. And so with that, I'm going to pass it over to Travis to tell you about beans.
Cool. So hopefully you guys can hear me. cool. Yeah, so thanks for the introduction, Antonia and Jared and everybody. So yeah, once again, I'm Travis Parker, and I have been working on the Scope Common Bean breeding project for the last several years, and I'll just be giving kind of a rundown of some of the releases that we have and some of the other things that we have in the pipeline right now. So next slide. Several years ago, at the beginning of this whole Scope program, we had some farms coming to us saying that they had this situation going on where they knew that there were really awesome varieties, these heirloom types, like what you see on the left here or in my background, more or less. And they knew that chefs were willing to pay a premium for these because of the higher culinary quality. And they knew that with increasing internet sales and whatnot, that they were able to get these directly to consumers who really wanted them and were willing to pay more for them pretty easily. And that was all excellent, but they knew they also didn't have the field performance that a lot of the main commercial varieties have, especially when they're grown on organic agriculture, which, as we know, is limited in certain nutrients sometimes, and whatnot, definitely a different production system. So these organic farmers were coming to us saying, how can we maximize like the resource use efficiency of varieties and kind of combine the best culinary quality with this better field performance? So we as breeders thought this was a really awesome opportunity, and that's kind of how it began. Next slide. We used two methods to get some improvement in these lines. So I won't go into too much detail, but basically with the first one, it's something called recurrent back crossing, and we are making lots and lots of crosses with the goal of having just like maybe like one of the great grandparents, something different with maybe a disease resistance. In this case, it was for bean common mosaic virus resistance. And so we mostly have something that's very similar to that original heirloom because maybe, the ancestry is 63, 64, it's heirloom, but we can, by careful selection, and this is all just traditional cross-pollination and choosing the best varieties that inherit resistance. By doing this method, we can get something that often is much better in the field because it doesn't have disease problems. That's one method. The other method is what we call the pedigree method. We can just make a single cross and we get this incredible amount of variation out of that, like what you see there. Those are all the F2s on the right side are all from one cross. Yeah, thank you. So just this huge amount of variation, that's just in the seed appearance, but we also get this tremendous amount of variation in the field, and we can make selections because a lot of these aren't very efficient in their use of resources on organic farms, but a lot of them are very efficient and even sometimes better than either parent. And so that's really what we want to use. That's what we want to see grown by organic farmers. So we developed three releases from each of these programs. So kind of a blend of both of them. Next slide. Once we had things that we knew were very promising, they were stably resistant to the virus or much higher yielding on our farms in Davis. Then we did 2 years of multi-location trialing. You can see the trial sites there. There's a gradient of temperatures and environmental conditions across these. It's very cool on the California coast, very warm in the interior. And then we had some things that did very well after this multi-location trialing. Next slide. You can see we did this in partnership with the Organic Seed Alliance. So this is Steve Peters. Some of you might know him. This is us doing a collaborative harvest out in Pescadero. Next slide. And you can see that what's coming out of these fields is very different. But on the left, we see one of the UC Davis selections. That's UC Southwest Gold. You can see that it matured very uniformly. We have lots and lots of seeds. On the right, this is the pre-existing heirloom parent, and we have inconsistent maturity and lower yields. This is just what's coming out of the back of the harvester. Next slide. And so we can quantify this a little bit more, and you can see that in each case, we have pretty major yield improvements. Again, this is all done on organic farms exclusively. So you can see that some of these are just much more efficient in their use of those resources that they're given in terms of water and all these things, often by 50 to 60%. Next slide. Again, once we had things that had passed that yield evaluation on multi-location trial farms, then we put those into taste tests. And this was kind of like the last set of selections, where basically we wanted to make sure that we maintained the high culinary quality of the parents. And so all of our selections were types that did have the culinary quality that was equal to or superior than the original heirloom parent. Okay, next slide. Cool. So these were all released in 2020. We've sent them out to farms who have been increasing them. So they were grown, I think, on 7 states and at least 3 countries in 2020, and then more in 2021, including in Europe and some places. Next slide. So these were all released to the public. So anyone is able to do whatever they want with the seeds. We don't have any intellectual property protections. It's all out there. And then if you're interested in the details of the ancestry of these types, or how we did our selections and things like that, we do have these registration papers, so feel free to check those out. Next slide. This is an example of some of the seed increase that we've been doing. So, for example, actually 2 years ago at this conference in Oregon, I was talking with some of the folks from Ernie's Organics in Idaho, and they were really interested in the varieties. So I sent them seed, and because of Idaho seed import permit restrictions, it had to be like less than 1 pound. So every bean here represents like 1 pound. So the first year they had 1 pound, they planted that out, they got 30 from some of these varieties. And then after the second year, 2021, they had 700 pounds. So you can just see like the seed availability is really expanding. Next slide. Here was a grow out that we did in Davis for a private company of one of the types. So everything that you're seeing there is one of the new varieties. Next slide. And if you're interested in getting seed, one collaborating farmer that we've worked with for a long time is named Mike Rieske, mrieske@aol.com. He's been working with us since the very beginning, and he was managing trials. He did tons of work. I mean, I would just send him seed, and I would go visit maybe like 1 time or twice during the field season, but he did like almost all the work on his own farm. So that was great. So if you're interested in getting seeds of these and trying them out, he's a great person to contact. But there are, like I've been mentioning, there are other folks that are growing this stuff. So feel free to ask if you have a preferred organic seed source. Next slide. So that was kind of our first generation, but it's hard for me to stop making crosses and making selections, because once you get started, there's never like an end line. So I've continued to get new BCMV resistance, those virus resistance, into different types, and trying to improve yields, trying to improve some other things, growth habit. And then another thing that's been kind of neat lately is I've been working on improving the pod shattering of some varieties. Next slide. So maybe some of you are familiar with this type. This is called Tiger's Eye, and it's highly shattering susceptible. We did come out with a shattering or a virus resistant type, but we didn't make any selections for pod shattering initially. So now I've been working on that. We have some promising types that are virus resistant and shattering resistant, but not yet released, but soon will be. And then cool. Yeah. Then these are some of the things. These are what I call pocket beans. Anybody who grows beans know what pocket beans are. It's just like at the end of the day, in fall, you go home and you're like, this is what ended up in your pockets as you were walking around the field, just cracking open pods and seeing what you have. So these are all things in the breeding program and or just like in our field. So just other things we've been making selections on lately. Next slide. The last thing I wanted to mention is that we also have a scoped sweet potato project that has been in the works, and you can see maybe at the top left there are some of the more traditional types, the kind of the white flesh types, the orange flesh types, They're really productive in our areas, but then some of these purple flush types have been getting really popular in the supermarkets around here. They just tend to be, at least in our conditions, much less productive and whatnot. So one thing we're trying to do is improve the yields of those types and improve the culinary quality. So we've been doing taste tests. You can see that at the bottom left, and you can see some of the results of the flavor score in the bottom center there. So that's just something else that's in the works. And especially if you're in California, and if you have any experience or interest in growing sweet potatoes as a grower, that would be, we're looking for people to collaborate with. So definitely feel free to reach out to any of us in the Scope team. I think that's it. Cool. Yeah, I just want to say thanks. I've had a ton of help from my PI, from the other Scope groups, from OSA, from a bunch of farmers and undergraduates and all kinds of people. So, and my funding sources, of course, as well. So there's lots of people to thank.
Cool. Thank you, Travis. So hi, everybody. My name is Will Hazzard. I am the student project lead for the Zinnia project. So some of you may be asking why Zinnias? Why did we choose a common ornamental flower for our breeding project? So before this even really began, we did a survey with a lot of our partners who we work with, who have done trialing for us, and we asked them, what is something that you would really like to see improved? And the number one thing that they really came back to us with was zinnias. Zinnias are really, and cut flowers in general, are a really emerging market for many organic farmers, particularly here in California. And something that makes zinnias particularly special in that regard is that they have very localized production. A curious fact about zinnias is that they don't like to be refrigerated. So for those of you who are familiar with commercial cut flower production, a lot of those are imports from Central and South America, or maybe Florida. And they're usually cut in the field and then immediately refrigerated and transported to florists all over the country. Zinnias, you can't really do that with them. So there is this really great opportunity for growers to, make money through events, their CSAs, and providing to local florists. There's a really growing interest in local flowers. In addition, we looked at zinnias because they are really important for our teaching and education. As Antonia mentioned before, you can look at the variation in zinnias. They're bright, they're colorful, and you can really get excited about them. In addition to that, they are a word called an obligate outcrosser, meaning that they don't really like to be self-pollinated. They like to be pollinated with other flowers, which presents certain challenges for breeding, but also many opportunities for learning as well. And then finally, we wanted to kind of expand our kind of network here in Davis of people we work with. So this has become kind of a joint project with the UC Davis Student Farm Flower Project. And the Flower Project teaches students, undergraduate students, about flower production, and they also provide flowers for local events at the university and throughout the Davis community, as well as providing a flower CSA. So really a great way of giving back and participating more in the Davis community. Next slide, please. So what are our goals? First and foremost, with ornamental breeding, novelty is really the key thing. So we are looking for novel color phenotypes. That can mean colors that aren't necessarily as common and you probably wouldn't see, in your local garden or maybe at your farmer's market. And the big thing for that is we're looking for intermediate color phenotypes. When you think of a zinnia, I think of, a big, bright, in-your-face flower that really stands out in the middle of summer. And while that's really amazing, it is not always the best flower for florists. We talked with our collaborators, with people who host events, and they want smaller flowers that have more of these blending tone colors. Your blushes, your soft yellows, your pastel colors, so things like that. Another example of improving that novelty is through color and shape, petal shape combinations. Multiple colors on the same flower, different shapes, so curled leaves, or double petals, things like that, making unique combinations that haven't really been available before. Improving resistance to powdery mildew. Most growers are really limited in their field season just by this one disease. So if we can improve the resistance in the flowers, we can really expand the overall field season. Stem length and flower size are really big quantitative traits, making the stems longer, better for florists. Smaller and larger flowers, depending on the variety, to make them a little bit, stand out a little bit more. And then finally, face life, so that these flowers will live longer in the field, in transport, and then they just look better when they are sitting there in the vase in your home. We are also trying to add some self-fertility into zinnias to improve some of our strategies for breeding. Next slide, please. So how do you breed zinnias? So when you go to your local seed supplier and get a packet of zinnias, usually those are sold in what are called multi-line mixes. So those are a great genotypic diversity, so lots of gene diversity in that packet you buy, but most of them look generally the same. So that's kind of the orienting goal for us in terms of how we've developed this breeding program. We are using what's called a full and half sieve population method. So we are making crosses, developing these families, and then trying to make these diverse families look generally the same for the purposes of eventually releasing a variety. And then we do that through pedigree mating. So we have a pretty sophisticated pedigree system in place. So we know what flowers came from which families and make sure that diversity is really, really high so the population can really thrive. With that in mind, we developed these four kind of idiotypes. So overarching goals that we are selecting for. They are improved queen line, so an improved flower with green and multicolored petals that has a lot of disease pressures and poor stem size. Bicolor cactus, so that multicolored flower with a cactus flower phenotype, which is a curled petal, and I'll show you some pictures of that in a moment. Pastel intermediate, which is again those light or blending colors, and then smaller Benaries. So Benaries giant is a really wonderful variety, great disease resistant, great stem length, but the flowers are just a little bit too big for a lot of florists to use effectively. So we're trying to select for smaller flowers that might be better for those florists. And as you can see in the bottom here, here's just kind of an example of what happens when you cross a zinnia flower. We have this lighter colored flower with this really bold bicolor in the middle, and that produced these softer bicolors, which is like a neat little mix. So even in that single generation, you can really see that you're doing something with these flowers, and it's really, really exciting. I can say that from personal experience. Next slide, please. So what have we done so far? Scope, this is probably currently the newest project in Scope, and it's something we've really had to build from the ground up. There is not a lot of protocols or guides out there for seed saving, for how to cross these, how to manage them effectively in a breeding field. So, as I say a lot, we're riding the bicycle as we build it. So we began by evaluating all the available germplasm out there that we could really get our hands on, both from commercial nurseries, seed suppliers, and the USDA Grand Germplasm Center. And we just did a big field evaluation, measuring the colors, the sizes, things like that. Then we spent a long time developing all of these techniques for crossing and seed saving. We made several hundred crosses between those initial 40 or 50 zinnia varieties that we got. And then we selected them and evaluated them with those 4 breeding goals in mind. We grew them all out. We had a really wonderful field season last year and evaluated those F1 families. And currently we are in the process of planning for our 2022 field season. And here are some pictures of Laura and another one of the grad students who worked with, Amy Groh, doing some crosses, and that's me measuring flower heads there at the bottom. Next slide, please. Here are just some great examples of what we've been able to produce in the field. You can see here, if you see something with a bunch of letters and numbers, that's one of our breeding lines, and something with a more distinct name is what we call a check. So the commercial varieties that we're kind of comparing them to, there's that queen lime that I was talking about, those really cool green flowers. Green's probably my favorite colored flower, so I may have a bias. The cactus chrysanthemum down there is that cactus phenotype I was talking about before, and something we really like. These are kind of the intermediate colors we're looking for. You can see a lot of shapes and sizes, a lot of diversity in there. Even in an F1, which in an inbred population, you'd expect that to all look the same, but we're working with things that are just full of diversity, even from the get-go. And something really cool we do with the program is that we use open pollinated varieties to kind of keep the diversity high in our breeding populations. So in the bottom corner, you can see this split between the yellow and red flowers, but those all came from the same family. And we've gotten some really, really exciting stuff just from letting the bees do their job and finding really interesting things that are lurking in those populations that are kind of unseen. Next slide, please. I'm going to show you also a little bit of the data we collected from this field season we had. This first chart is a distribution of all of the colors that we actually saw in the field. This is from about 1,000 plants. On the chart, most of these colors correspond with what they look like in the field, and there is a small key over on the left-hand side. And if you see those two numbers, so that end with all the little stubs at the bottom, that's a multicolor or a bicolor phenotype. As you can kind of see here, it's really interesting. We know a little bit about the way color is inherited between flowers, and red tends to be dominant. So you can see red, orange, and pink and magenta really made up the majority of what we saw in the field, which is really interesting, from a biology perspective. But not everybody loves red flowers, so it presents a certain challenge and a need for planning when we're advancing things from one generation to another. Next slide, please. I've got a couple slides on what stem and flower size distribution look like. So you can see there's this really nice normal distribution for stem length. So all of them are about little over a foot long, and we put this through a model and estimated the heritability, so what one will look like from one generation to another, and it's pretty high. Heritability is between 0 and 1, and we got 0.79 for stem length, which is really exciting as a breeder. It means we can really make some improvements. And a normal distribution means there's things that are small, things that are large, and things in the middle, and we really think we can do some really cool selections on them and really get some advances there. Next slide, please. Same thing kind of with the flower diameter distribution. Again, that normal distribution, great opportunities for selection, and a really, really high heritability. That means we could get some really, really great responses in the generations to come. Next slide. And finally, something that's interesting I just wanted to point out is like looking at this comparison between flower size and stem length. So you can see here, this is the means of all of the families kind of plotted on this graph with their stem lengths and their versus their flower diameters. And there seems to be somewhat of a correlation, meaning that the big taller plants tend to have bigger flowers and the smaller plants tend to have smaller flowers. This is really interesting, kind of an opportunity for data and intuition to appear data, but also it will present some challenges since we're trying to have something with small flowers and a big stem. So not always great news when you're developing a breeding program. Next slide, please. And finally, there is our base life tests. So we have been developing a series of vase life experiments for the Zinnia program. We've gotten kind of two big experiments. We call it the rough and ready, which is where we just took flowers from every family in the field and put them in vases and then counted how long they lived. But we wanted to do something a little bit more controlled. So we did a well-designed, controlled experiment, and you can see the data from that in this box plot over here. And every one of these represents a different family in the field, and those lines in the middle are the mean vase life. As you can see, there is a significant difference between some families versus the others, which is really, really exciting, meaning that we can, simply by performing selection over a few generations, we can really get a great return, not just for our growers, but also for consumers as well, making like a zinnia that will really just stay on that shelf for forever. And I believe next I am passing it over to Laura.
Yeah, thanks, Will. I'm going to be giving y'all an overview of our wheat project today. One of our students, Danyelle, was going to present this section, but she had a schedule conflict. So I'm going to be talking instead. Danyelle and I work alongside our wheat lead breeder, Allison Krill Brown, which I think she's in the audience somewhere. She and Danyelle and I manage the Scope wheat breeding project. The objective of our project is to develop small grain varieties for California organic growers and artisanal food producers that combine improved yield, weed competitiveness, and disease resistance with unique flavor profiles and quality characteristics. In terms of organic wheat specifically, some important things are weed competitiveness, early season vigor, and yield in dry farming conditions. In California, organic wheat is usually grown over the winter, so we can take advantage of the winter rains. But even with some winter rains, we've had a very dry month. We hadn't had any rain in January in our area. So we need varieties that are adapted to having little to no extra irrigation. And in terms of grain quality, protein is really important for wheat overall. But specifically, the quality of protein is really important for organic wheat, since organic wheat usually has a lower percentage of protein compared to conventional wheat. But we like to say that there's no such thing as a bad level of protein. So usually high protein is better for products like bread baking, while lower protein wheat is better for things like pastries. And we like to say that there's a place and a product for every single kind of wheat that we can grow. Color is also something that's really a really important trait we work with, because a lot of the millers and bakers we work with are interested in not just organic flour, but whole wheat organic flour, which better highlights the difference in colors between different varieties. So that gets me into our breeding goals. Our first, and I think one of our most exciting breeding goals, is to breed colored wheat varieties, such as blue and charcoal and yellow varieties, by incorporating these high anthocyanin and lutein traits into populations to increase their antioxidants. So we currently have about 100 populations at the F5 to F7 generations. So that's 5 to 7 generations after we made the initial cross that we've been selecting. And these are also being tested specifically for whole wheat baking. So we can really see those color differences between the blue and a typical red or white wheat that you would find at the store. Our second breeding goal is to make crosses between heritage varieties, which are really valued for their flavor profiles, and crossing these with modern varieties, which have improved yield and disease resistance. We have about 27 populations at the F4 to F6 generation. And finally, we're evaluating varieties which have expired plant variety protections. So these were bred by other universities, other groups. And since the PVPs have expired, we've been doing a seed increase and evaluating them specifically for organic growing in California, since they were never tested under those conditions when they were bred. And so we started with 78 varieties from the USDA germplasm repository. It's called GRIN. And we narrowed these down to a list of about 22 varieties based on how they performed for us. And so we've been releasing these for on-farm trials. We've been doing trials not just at our farm, but in a few different locations. So for each of these 3 breeding projects, we're looking for all those important traits that I was talking before about like wheat competitiveness and yield, but we're also trying to evaluate the quality and the flavor of our lines. So we work with the California Wheat Commission to evaluate our wheat for its baking quality. 2 tests that we work the most with are the mixograph test and the SDS sedimentation test. And so the sedimentation test is located here. You can see it's a quick and simple procedure that's a good indicator of protein quality. So it's based on the density and swelling capacity of gluten-forming proteins. So we add a little bit of flour into a test tube with an alcohol acid solution, and we let it settle. And you can see the amount of sediment is a indirect measurement of the desired protein content. So higher protein or better quality protein usually has a higher level of the sedimentation that you can see at the bottom. And you can see some of these blue test tubes here. Those are actually tests we did with our blue lines. And so you can see that the anthocyanins dissolved in the solution. And then we also have the mixograph test, which you can see here. It's basically like a mixer with 3 prongs. And it produces a mixing graph like this one. And so it shows the optimal mixing time. So it'll show you the maximum. And then additional mixing will break down the dough, and you can see the curve at the bottom. And so the rate of the breakdown is associated with baking quality. So you want to have a slower breakdown. Another key component of our quality testing is bake tests. We do a few different types of bake tests, including the straight dough method, pouliche and sourdough, and also some other baked goods like tortillas. We've also done waffles. The straight dough method is just using baker's yeast. The pouliche or sponge method is where you make a sponge that you allow to ferment, and then you add that into your final dough. And then sourdough is, the method that became super popular at the beginning of the pandemic. You have a sourdough starter that you have to maintain, and it takes a lot more time. And we actually found that you can make a good sourdough bread even if you have really bad quality grain. That's the great thing about sourdough, is as long as you can get just the right measurements and the ratios, you can get good bread as long as you have lots of practice. So we actually found it's not as good for our purposes, because it takes a lot of time, and we can still make good bread. We have held several taste tests, both at home during the pandemic and on campus. So we had our students take flour home that we milled for them, and they made tortillas. So you can see in this photo here, we have a couple of different lines that they tasted. They made, I think this one's our yellow wheat. It's called UC Amarillo. It's a variety that we released in 2019. And we have a few reds and a white. So you can see, just even with the same baking method, you can see the difference in colors and the structure of the tortillas between the different varieties. And you can also see at the bottom here, this is a 100% whole wheat bun taste test that we've been doing, where we tested a red, a white, and a yellow, and 2 different milling techniques. One was hammer milling versus stone milling, just to see how the whole wheat rolls would taste different. So that's all I have for right now. I think next up, Lewis and James are going to talk about tomatoes.
All right, thank you, Laura. Hi, I'm Louis Salazar. I am a PhD student at UC Davis, and I am also the team lead for the Scope Tomato project, and I've been team lead since early 2020. So overall, the goal of the Scope Tomato program is that we're trying to produce new tomato varieties that are heirloom types, and primarily we're focusing on improved traits for specifically organic production as well as for the fresh market. We have an emphasis on heirlooms just because overall, I think a lot of people kind of see them as having superior flavor and whatnot. But as some of the other crops that we talked about, there are some specific challenges and organic production that we want to address in heirloom tomatoes. So some of these traits we're interested in are yield, flavor, disease resistance, color, shape, and size. Heirlooms are not only good flavor, but as you can see in this photo that we took a year or two ago from our summer field, they just come in all shapes and sizes and different colors. They're so unique and interesting to look at. I think people really want to see this in their tomatoes as well. So we also have a focus on how they look as well. And of course, we focus on things like fruit quality, such as blossom end rot, cracking, and sun scald. Next slide, please. So historically, the tomato program at Scope has used inbred line development. So primarily, we've used pedigree breeding to make our inbred lines. And this has been done using anywhere from like a 2-way to a 4-way cross, so 4 different parents. And typically, we will plant out these families and make single plant selections in our summer field. And since I've been team lead, we have narrowed our focus a bit in the tomato program, specifically in the inbred lines, one of which has been to stack disease resistances in an heirloom type tomato. Heirlooms are typically older varieties and have also been geared a little bit more towards things like home gardens. So often in many heirloom types, there's a lack of disease resistance. So in order to increase yields as well as to be more efficient in organic production, we wanted to be able to stack some of these common disease resistances in tomatoes. So we focus on things like Fusarium wilt, Verticillium, Phytophthora, as well as nematode resistance. And we've done this by using marker-assisted selection. In all our crossing and breeding methods, we use traditional breeding methods and crossing, but we do use marker information to make informed selections in some of our families in order to right away get the best allele combinations and to better and to efficiently use our resources at Scope. And we're pretty excited to say that we have a few families in development at the moment that actually have homozygous resistances to all the major suites of resistant loci that we're looking at with those diseases that I mentioned. And a main reason why we're very interested in having homozygous resistances to some of these diseases is that they would make excellent parents to cross with, and especially in hybrid breeding, which we'll talk about a little bit next. Oh, not done yet. The second thing that we're interested in is making a line that has very good flavor and also something interesting and unique about it, such as the color or the shape. In my time as team lead and as well as my undergraduate, I worked in a tomato flavor lab for 3 years. I've heard so many stories from people sharing how their parents used to grow tomatoes in their gardens, or their grandparents used to grow tomatoes, and they would be the most flavorful, like best tomatoes ever, and how disappointed they would be in grocery store or like some other fresh market tomatoes. And so this actually has been a main driving point for me in the tomato program, having this big focus on flavor and overall consumer likability. So we're aiming for another line that we think consumers will just be really pleased with. And this is another photo from some of the lines grown in the tomato program, showing just how unique and diverse the germplasm that we use are in the program. Next slide. So Antonia mentioned this earlier, but traditionally we've been doing inbreeding in the tomato program, but as of last year, we started with a hybrid breeding program. This is in collaboration with Polytechnic, as well as some other small organic tomato breeders. And we have a couple major goals with this hybrid breeder program. One is to compare the gains made with hybrid breeding versus inbred lines. So we definitely have made some progress with our inbred lines. However, as we'll discuss a little later, we've also have been met with some challenges. So we want to see what gains we can make and how quickly if we were to use a hybrid breeding schematic. And of course, we have things, what we kind of look for are again, yield, disease resistance, but again, still with a focus on things like desirability to consumers, such as like flavor. And this will also be a great educational opportunity in scope. Throughout the different crops that we've worked with, a majority of them have been focused on generating inbred lines. So this is a great opportunity for the students to have experience in hybrid breeding, generating these F1s. I myself only have experience in generating inbred lines, so I'm very excited to see what the outcome of this program will be. Next slide, please. For this hybrid breeding program, we, in collaboration with Cal Poly and other breeders, we've pulled together 23 lines of interest, also some lines from scope, and we grew them out in 2 locations last year. One being the UC Davis student farm, and also at Cal Poly Pomona, both of which are organic fields. And the main objective was to identify desirable lines that we could use in conducting a dialogue in order to assess, the combining abilities and to see how well or like how much gains we can make by producing F1s. So the major traits that we looked at were yield. We conducted a taste panel of the parents to see what tastes good and what we would like to see as a parent in our tomatoes, as well as looking at Brix. So Brix is a measure of soluble solids in fruit, and this is also a measure of basically soluble sugars in tomatoes. So it would act as a proxy for selecting on improved flavor as well. And in this photo was a planting from last year during the summer field. And so I'm going to turn it over to James, who will discuss some of the data that we collected from the field last year.
Hello, everyone. I'm James Weeks, one of the collaborators from Cal Poly Pomona. So from this year in 2021, we've had some interesting results and some unexpected results, which makes for an exciting collaboration experiment. And it's partially contributed kind of to this decision of moving towards the production and testing of hybrids. And between the 2 sites, we naturally observed some variation between the lines and the sites. We found a small correlation between yield at both sites. But we can also see where the arrows are indicating the parents we've selected to produce these hybrids. And we tried to pick things that have done well at both sites or really exceptional one or the other. The other thing to kind of notice, though, is between the 2 sites is this difference in magnitude of the yield. So at UC Davis last year, there's quite a bit larger amount of tomatoes harvested compared to Cal Poly. But another thing that really kind of stood out, it was one of the unexpected things I saw, was that CPP 46 Black doing so well at Davis. I mean, prior years at Pomona, it was just middling in terms of yield. We liked it for other years. So it was having the opportunity to test it at Davis and then see it perform so well. It's kind of astonishing to me, at least. And then 1733B2 was a top performer at Cal Poly. We've had a very harsh year at the Pomona site. So quite possibly there's a little bit of resilience in that line. Makes it interesting to use as a parent. Next slide. So for Brix, though, we've observed a much stronger correlation between the 2 sites, which suggests we can be a little bit more confident that between locations, tomato that performs well at one site is going to perform well at another site for Brix. And again, we can see those arrows indicating parents that we've selected to use for the hybrids. And they don't necessarily fall to the complete high end of the Brix, but we do see at 1733B and Magic Bullet are fairly high at 6% and 8%. So we kind of would expect their progeny to potentially have good Brix performance as well. And as Lewis mentioned, in addition to the Brix testing, UC Davis was able to form a tasting panel. Next slide. Which is again really useful after walking through the field, tasting hundreds of tomatoes. I think it's biased my perception of tomato flavor a bit, So being able to get some outside perspective is really great. And kind of see some interesting differences that occur between our selected parents, where Magic Bullet had 8% bricks, but now it has an average tasting score of 2, whereas 46 is a really high yielding tomato with very low bricks scored right in the middle. So what happens is we kind of see that these parents are spread out across all these different metrics. Some of them doing well for a particular one, but we go and we combine, hopefully their progeny, find some progeny with the potential to perform well across all these metrics. And then another, I guess, next slide. Another thing we're really interested in is the correlation between these traits. So how does bricks correlate with the tasting score? We see a small positive correlation to the two, which is good. We aren't causing serious harm to the flavor of tomatoes by selecting the bricks, but also suggests the bigger picture of tomato flavor is not encapsulated or explained entirely by the bricks. It could be individual components or ratios between the components within soluble solids that is maybe not being captured by the bricks data. So again, that tasting panel is very useful for making selections and decisions about what to incorporate into hybrid production. Next slide. Probably one of our strongest supporting results for the decision to move towards testing hybrids is this negative correlation that we're observing between yield and bricks. And it kind of highlights the difficulty to improve multiple traits at the same time, especially two fairly important traits, bricks and yield. So it's resulting in having to adapt, re-strategize to kind of break this negative correlation. Next slide. So another part of the collaboration is understanding and adapting to the variation between the sites, accounting for this in how we plan things. And these two graphs kind of show the variation at different sites of bricks. Whereas at the Davis site, you see variation for bricks for each parent is a little bit larger, whereas at the Pomona site, it's compressed. So there's potentially explanations for this, but we're aware of this going on. It's something we consider when making selections. Next slide. I think overall the collaboration has been really an excellent shared experience. We've had similar challenges both sites, turbulent pressure, some extreme heat events, difficulties arriving from dealing with COVID, delays in material supplies. But once collaboration has really shined through and has led to some successes, first off, allowing us to deal with those challenges, but also introducing digital bricks testing, disease marker testing, and excellent. Those are things that aren't always available at Cal Poly. Then the ability to share resources in germplasm is critical. We had a greenhouse wiped out early on by mite infestation, but I was able to replace those plants within a week through the collaboration. That's just excellent. So the replication cooperations really increase the resilience, overcoming those challenges. And it's additionally provide a lot of excellent learning experiences, opportunities to exchange methods and techniques and knowledge, all while working on some real world plant breeding problems. Also having multiple sites is allowing us to estimate environmental variation and maybe consider adaptation for a wider area of California sooner in the process. So then one of the things we have been doing a lot of at the site is photographs and video recording. So one day I was out in the field recording field notes and managed to generate 20,000 photos. And later in the year, I generated a few hundred photos of fruit from different plants. But I think one of the things to look forward to is possibly taking all of these photos and turning them into useful data points. So I think we're happy with a lot. There's a lot to look forward to. like to hand everyone's attention back to Laura and the peppers.
All right, thanks, James. All right, I am going to end our presentation by talking about 3 pepper breeding lines that we have ready to release this year. Hopefully you guys will start seeing them in a seed catalog for next field season. So our pepper breeding project is focused on breeding jalapeno peppers and bell peppers. The jalapeno breeding project was initially a teaching project that, alongside Travis's bean project, helped start the overarching scope project. And our jalapeno breeding project is focused on breeding jalapeno for jalapeno poppers. And our bell pepper breeding project is focused on breeding bell peppers for reduced sun scald. And I'm going to talk about each of the projects separately, and I'm going to start with the jalapenos first. So our jalapenos, if you don't know what a jalapeno popper is, it's a common bar food or party food where you stuff a jalapeno with cheese, you bread it, and you deep fry it. So really tasty food, but we thought, hey, why not add a bigger cavity so you can stuff more cheese into your delicious jalapeno poppers just in time for the Super Bowl. Woohoo. Okay, so by enlarging the cavity, we can stuff more cheese, but how do we achieve that? We want something that has a thick cavity wall. We want something that is moderately spicy, something that has an equivalent or higher yield to our checks, and also something that's a little bit wider and a little bit longer to help increase that cavity size. Our main check variety has been early jalapeno, which is an open pollinated variety that a lot of the organic growers that we talked to said that they plant this. And our 2 breeding lines we are calling CK and CJ. So CK is a little bit spicier. It's a little bit longer than our Czech variety, and CJ is a little bit more mild. It has a thicker fruit wall, and it's not quite as long, but it's definitely a lot fatter than our Czech variety. And so in order to get these lines, we crossed bell peppers with jalapenos, jalapenos with jalapenos, and then we made selections over, I think, about 5 to 10 generations for each of these. And we achieved both of these breeding lines that have good yield, sturdy cavity wall, and different levels of spiciness. And so since the past 3 years, we found that the width and the thickness of the fruit walls of both lines are significantly different than our early jalapeno. They're much longer, or they're much wider and much thicker. And the CK line is definitely much longer as well. In addition, our CJ line over here is a little bit of a later season variety, while the CK line is not quite as early as early jalapeno, but it's more of a mid-season to early season variety. We didn't find a significant difference in the yield between the lines across the past 3 years, which is good. It means that at least our variety performs just as well as one of our comparisons, but it doesn't perform better. Part of this is we don't have a lot of resources to do many replications and multiple location trials that statistical analysis requires, but at least it's promising to show that, hey, we have something that's a pretty good line here. We also evaluated the quality as jalapeno poppers. So like all of our other projects, except for the Zinnias, we've been doing taste tests. We did participatory taste tests at home in 2020 and 2021 due to the pandemic. And we had an in-person taste test this year or in 2021, and then also the first one was in 2019. And so we partnered with chefs from the campus dining services to prep the peppers as jalapeno poppers and to rate them for things like taste, overall flavor, level of spiciness, appearance, stuff like that. And we didn't find a significant difference in the flavor between any of our 3 lines, but we did find a slight positive correlation between the spiciness rating and the flavor rating. This makes sense because people who attend or volunteer for a jalapeno popper taste test probably also like spicy food. And so they're they tend to like things that are a little bit spicier than maybe someone who wouldn't go to a taste test would. And this is a graph just showing the means and the distribution of the taste ratings for the jalapeno poppers each year. You can see these dotted lines here represent the mean for each line at each year. And they're pretty similar. There's not a statistically significant difference. You can see the blue shaded area represents the standard error. But it means that our lines have a pretty similar taste rating to early jalapeno, but they're a little bit bigger, so they're better for jalapeno poppers. And especially with our less spicy line, it's good for people who like to eat jalapeno poppers, but maybe don't like the spiciness as much. They just want a little bit. Okay, and now I'm going to switch gears and talk about our bell peppers. The goal of our bell pepper breeding project is to produce a bell pepper with reduced sun scald. So sun scald is an issue, like a cosmetic issue with fruit. So it results in papery or yellow patches on fruits. This also happens in tomatoes, not just peppers. And it causes a yield loss because you can't market these peppers. So our goal was to increase the marketable yield by reducing sun scald. So our variety here, CI, not only has reduced sun scald, but is also slightly sweeter as a green pepper versus our comparative lines. A lot of people don't like green peppers because they think they're bitter, they don't taste very good. But ours, out of a small survey we did, ours rated slightly higher in terms of flavor versus Golden California Wonder, which is an open pollinated variety that we compared it to. Something else interesting about our fruits in terms of shape is that it's a little bit shorter. It's about 20 to 28% shorter than our comparison varieties. So it's, instead of being like that long rectangular shape that you see bell peppers, it's more of a square shape. And it also has more lobes. So you can see in this photo here, we have a lot of fruits that have like 4, 5, even 6 lobes at times, while most pepper varieties tend to have 3 or 4 lobes. So this is something that just makes it have a different appearance compared to the typical bell pepper. And okay, so finally looking at some yield data for our bell peppers. Consistently across the years, our line has had lower sun scald compared to our Czech varieties, even including Red Knight, which is a F1 hybrid. We also, and this is despite some issues we had over in 2021. So for example, at Full Belly Farm, there was a labor shortage in 2020 due to the pandemic. They weren't able to put up the shade structure that they normally put up. There was also a lot higher weed pressure because they weren't able to maintain their normal weeding schedule. And despite this, our line still had a lot lower sun scald and still had a higher yield than either of the Czech varieties on a per plant basis. And also last year was a pretty ideal year in terms of conditions. We had the shade structures up, there was really great weeding, everything went well. And of all the plants that we harvested, we didn't have a single fruit lost to sun scald from any of the harvesting we did for our line specifically, even though the other 2 lines had maybe equivalent to 1 or 2 fruits lost per plant due to sun scald. Additionally, we had some issues with at Hardy Fork Farm. We changed locations on the farm between 2019 and 2020 and 2021. And so you can see a dramatic change in the yield from 2019 versus 20 and 2021. And despite this, our line still performed better, or at least similarly to our lines. And it kind of complicates things because we had a really great year in 2019, and it was just really hard for us to get anything that was marketable at all. And so even though there were there's fruit on the plants, they were all so small by the time we delayed harvest as long as possible, they just weren't marketable. So that's it for our presentation. I wanted to thank you all for coming and listening to us. If you want to contact any of our presenters, you can check out our emails. We'd like to thank all of the people who have helped our project over the years. Not all of them are on this list, but We also, if you want to check out our project more, here's our website, and we also have our Instagram page.
Great. Thank you so much, Laura, and everyone else for that fascinating presentation. Thanks to Antonia. Most of the questions people have had in the chat have been answered. I will, but if you have more questions, you can feel free to do one of two things. You can either use the raise hand reaction, or you can type it into chat. And but the questions that I see that I don't think have been answered yet. Bill Kemling asked, what Scoville scale range are the poppers in?
I can answer that. We actually haven't tested it. We haven't done that before. We don't have the machinery. I don't know how to do it. I would be looking to learn how to do that.
Yeah, I mean, in my experience, it seemed like the one line we could eat in the field raw and it was still spicy. But the one that everyone liked a little better in the taste test because it was hotter, that gave me the hiccups right away. So. It's not a Scoville measurement, but
Hiccup measurement.
Yeah.
John Pattison asked, in breeding work, how do you know when the desired improvements can come about simply by changing management practices, for example, timing, fertility, water, etc., versus improvements that require new genetic changes in actual breeding, especially for traits like yield, flavor, nutrition, vigor, disease resistance?
That's a good question. I think the main thing to figure that out is doing one, trial year or location after another. And there's definitely always improvements that you can make with fertility, watering, your planting time. But there's always sort of a range that a given cultivar is going to have. Like it's, genetics won't allow it to go further one way or another. So, but that's why we do a lot of trials. And I don't know if anyone else has comments on that from the group, but.
Can I jump in on that as well?
Yeah, please.
So it's, in the question, it says simply by changing management practices. I think one of the big powerful things about plant breeding is that you can breed something once and then never need to do that. If you can reduce your number of weedings by one throughout the season by having something that's really vigorous early on, or whatever it is, there's so many different traits. If you can send a crew out one time less, that's huge, because then so I guess from our point of view as breeders, it's I know with at least one of my varieties, I'm like, oh, if only I had gotten this one extra gene variant in, we would never have to deal with X again. So from my point of view, one of the powerful things about breeding is that it allows us to simplify the management. So basically, from growers, we always want to know how would you like to see management simplified, and then we can breed for that. And you'll hopefully not have to deal with it year after year after year, field after field after field.
Yeah, and for some of the disease resistances too, like what Travis is working on and we're trying to do in the tomatoes, there's not a real management way to get around some of these things, and they can be seed borne. So it's a compounding problem, but we can put these disease resistant markers in there just by traditional breeding, and then that solves the problem for the long term, which is great.
Yeah, especially in organic, when you often don't have another solution. We don't have the tools of just spraying just to get rid of a disease problem that you would have in a conventional system. And so one of the only tools we might have is to breed in disease resistant traits.
Thanks. Riley wants to know do you hand pollinate your peppers to cross them?
Yeah, that's how we did our initial crosses. But we actually do have a big issue with cross-pollination in the field. Peppers are mostly selfing, but I think it's like 70 or 80%. So there's still a pretty good chance that they can outcross, especially in an organic environment where there's a lot of pollinators floating around. So we have to cage our individual plants to make sure that they don't accidentally outcross. We actually had that as one of our jalapeno lines is actually technically a bell by a bell cross, but it's clearly not because it's spicy. So it must have outcrossed early on in the project when we weren't caging our plants.
Thanks. Caroline asks, I work at a university farm that primarily serves undergraduate students. From your experiences, what are the best ways for undergraduate students, with or without backgrounds in science and research, to get involved in plant breeding projects?
Well, I think just having the plant breeding projects housed at our student farm has helped expose the undergraduate students to the whole concept of plant breeding. And a lot of times, actually, the undergraduates that we've had get involved in scope are a real mix of interests. Like we've had, a lot of students come to us from the sustainable ag and food systems major because they have to do internship requirements. They have a very broad set of interests, but like this summer, one of our students, he's a math and linguistics double major and just wanted to, be outside and get to know and understand plants more. And it's really broadens, I think, our ability to see things, when we look at them through students' eyes with different kinds of backgrounds. So I think just having the opportunity there. And then also, since Laura has started with the project, we've really been able to focus more on shaping the student internships so that we can get the students more involved in how to analyze the data once it's collected. We always have them in the field taking measurements and observing and things, but we've been able to sort of involve them on a deeper level. Laura or anybody else, do you have other comments?
Yeah, I mean, I was going to say that our breeding project was started by students. So it's if a project doesn't exist, find someone who might be interested and then make one. So if you have like a student farm or some kind of experimental farm at your university, or like a community garden or something, or just a group of people who are interested in gardening, you can start your own breeding project. It can just be small, like pick a crop that you're interested in and make some selections, grow out different varieties, see what performs well. Like it doesn't have to be a formal program. Like I think that ours has become over the past few years.
And I would also add that we often kind of advertise the scope program at courses at Davis. So either like plant genetics or like general plant sciences courses, we often attract a lot of students in that way. So if you have a project going on already, or once you form a project, it'd be a good idea to advertise, have a few people speak at some courses or classes to help advertise the project to recruit students.
Great. Kevin wants to know, how do we request seeds?
That's a good question. Travis gave some links there in his presentation to some of the people who are growing out his seeds. That's been a little bit of a bottleneck for us at UC Davis. We don't really have the capacity to, do packet sales once we have a finished variety. Of course, if you write to us, we can do our best to send you some of these seeds. But ideally, we'd like other people to take on the seed production and sales. I don't know, Travis, if you want to comment more on that. He sent a lot of of seeds out over the last year or so.
Yeah. Well, I would say that other than common beans, I don't think there's been anything that's been released yet, but there's obviously some stuff that's coming up. So really it is still like property of the breeding program.
Right.
And it's not really something where we're supposed to just be like giving it all away until it becomes public and it's totally released. But when we were doing our trials, we were doing material transfer agreements. So, there is a mechanism, but it can be a little bit trickier. So depending on what your objectives are, like if you want to be doing a participatory on-farm trial, I would say maybe go with the MTA route. Otherwise, we'll do our best to keep everyone up to date on the new releases. That's something that comes to mind for me. I don't know if you wanted to say anything else, Antonia, about that.
Yeah. Thanks for mentioning the material transfer agreement thing. So yeah, we're often looking for farmers to work with for on-farm trials, but because of the way the university works, we have to have this like legal agreement set up. Basically, that just allows us to have a publication at the end, because these are going to be public releases anyway, but that's sort of what is getting protected there, I would say, really. And like the peppers, we're going to be having a joint trial with Siskiyou Seeds this year, and we're working on wrapping up the MTA with that. So if they like the peppers, they might end up putting them in their catalog, for example.
Great. So Kevin is following up, saying we could do all that here at Johnny's. Who or how do I get started?
If you want to get in touch with me, we could work on that. I feel like we've had some different attempts to work with different seed companies over the last few years, and many of them have fallen through the cracks. So that would be great. I'll put my email in the chat.
Wonderful. Does anyone have any other questions for the team of presenters here? If not, yeah, do any of you have any closing remarks? Well, I would like to thank all the presenters, and I would encourage everyone else to do the same. You can either come off mute or you can use your hand clapping. This was really an interesting and informative presentation, so thank you so much.